Bridget Ogilvie. I'm a biological scientist, of course, and my interest when I was doing research was in the immunology of infection. And pursuing that, I worked at the National Institute for Medical Research for 17 years on the staff of the Medical Research Council. Then I switched careers and joined the staff of the Wellcome Trust when it was quite small. Stayed there for 19 years, ending up ultimately as its director. And I've been retired for a number of years, occupying myself with a variety of non-remunerated roles. Well, I became a scientist not because of any individual, because I grew up on a sheep farm in Australia and saw the power of science to transform agriculture. Before my very eyes, the farm became more prosperous because of vaccines for sheep against the bacterium Clostridium, and then we had all sorts of parasites, and initially the anthelmintics were very poor and they got better and better. So it was practical experience that made me become a scientist. I'll tell you a moment that I'll never forget, which shows the persistence of parasites. I was out with my father on our horses, and we were moving a mob of young ewes which were heavily in lamb for the first time from one paddock to the other, and suddenly one of these ewes that looked splendid just dropped dead, just like that. And my father said, "Oh, I think they've got fasciola hepatica, liver fluke." And he always carried things so he could investigate deaths or something that was happening on thing. They opened up the sheep and it had no liver. It had just, its belly was full of a mass of tiny liver fluke. Which, and all sheep got liver fluke over and over again. There was no immunity to it at all. But when I was a child, we used to lose sheep from bacterial infections. Along came the vaccines, we no longer lost them from that. And it had a deep effect on me as a child, because I spent a great deal of time on the farm from the age of 6, by which time I'd learned to ride a horse, until I went really to university. So I saw these changes, which had this huge effect on me. And then, of course, the people who inspired me were the dean of my undergraduate faculty, who's a very casual, enthusiastic vet. And then eventually I went to Cambridge and did my PhD, which was, it wasn't a major experience for me actually. Then I went to the National Institute of Medical Research and joined the staff of the Department of Biology. And at that time, immunology was a very powerful discipline at the National Institute for Medical Research. And the people there were really the founders of the discipline. Up there was Peter Medower, who's the director, had a big influence really through his fabulous writing on me. And then there were people like Anne Mitchison, John Humphrey, Bridget Askenas. There was a real focus for international training in immunology, which spread out around the world. So that was a huge influence. First of all, it was a wonderful scientific environment, very collaborative, very welcoming. Peter Medower, who was the director the first half of my time there, was a zoologist who was very enthusiastic about parasites and things such as that. And we were greatly encouraged to become much more experimental. Up till then, parasitology had been largely descriptive. And so we applied experimental analysis of what did the immune response mean in terms of the host-parasite relationship. And what fascinated me, as it had as a child, was whilst we had vaccines for bacteria for our sheep, the parasites kept coming back and back, and we had no vaccines. And so it always interested me, why is it that some infections escape the immune response and others are controlled by it? And of course now we know, when I think of what we were trying to do, our naivety about how the immune response and everything else, it was really mind-blowingly sort of elementary. But you have to start somewhere. And so all of us in the Department of Parasitology were pursuing this idea, why is it that infections are not controlled? And of course, one of the first things where people demonstrated this was with African trypanosomes, which keep varying their surface. So the immune response is forever having to pursue a new object, because they control the first wave of parasites, and then the second, one or two escape, which have a different surface, they grow up and then they're controlled. And then eventually that was shown for malaria, and of course for many viruses, not least of which is HIV. But that was a long time ago. I was really active as a research worker between 1960 and 1980. So, you know, I saw all this evolve, and I mean, it was the most wonderful time. Organised play for adults. That's what I always think of as my time as a bench scientist. It was enormously exciting and such fun, you know, it really was terrific, because there was almost every week there'd be something new which you could use immediately. But now I think life has got far more complicated, and you really can't work in that single-handed way, surrounded by people doing similar things. You really have to work in teams far more. And I don't think, quite frankly, it's quite as much fun.